Welcome back. So we are starting with an introduction to anatomy of plant cells, where we will discuss what are the different organelles of plant cells, what are the functions of those organelles, and then coming on to what are the different plant cell types which you can come across in a whole plant, and what are the different functions of those plant cell types or plant tissue types. So starting with the plant organelles, this you must have already seen. So what are the different kind of organelles we find in different eukaryotic organisms? You can find the major organelles are mitochondria, then plastids, endoplasmic reticulum, Golgi apparatus, then nucleus, ribosomes, then in plant cells or peroxisomes are also there. There are many micro bodies, then vesicles are there, and in plants you find vacuoles. So what are the different functions of these? Let's start. And moreover, in plants as well as in bacteria and fungi, you will find cell wall. Now the cell wall composition in bacteria and fungi is different from the plant cell wall. In what way? Any example? How is it different? What contains?
Fungi contains Ergosterol and not.
So talk in terms of plant cells versus the others. What plant cells have, the others don't have. So cellulose, then what else? Hemicellulose, lignin, pectin. So these are components of plant cell walls. Now plant cell wall itself is divided into primary cell wall and secondary cell wall. Now primary cell wall, majority of the plant cell types which you will find in the plant will have primary cell walls, all the plant cells, but not all plant cells will have secondary cell wall structure. So we will be talking in details, giving you some idea about how primary cell wall is different from secondary cell wall. Now what is cell wall? Cell wall is a component, it is a layer surrounding the cytoplasm or protoplasm. Now protoplasm is composed of cytoplasm and the nucleus. So everything of the cell apart from the cell wall. So the function of the cell wall is to protect the plant cell. Now depending on the various stages of the plant development, the cell wall composition modifies. Now let's talk about chloroplast. Now chloroplast, it is a distinctive feature in plant cells. You may find it in many macroalgae as well or algae, but in majority all the plant cells they have chloroplast. What is the function of chloroplast? These are nothing but modified which class of organelles? Chloroplasts are the organelles which we were talking about, it belongs to which class? Plastids. Now plastids, their pre-original form when it further differentiates into different kinds of plastids is proplastids. Proplastids when divides and gets a specialized function, then it becomes plastids. Now there are many different types of plastids. You must have heard about chromoplast, amyloplast, leucoplast, elioplast, proteinoplast. So depending on the specialized function they do in a plant development and growth, the name is there. For example, chromoplast is the most common which you must have heard. Chromoplast is apart from chloroplast, chromo, so where the colored pigments are stored. So now talking about chloroplast. Chloroplast among them are the most abundant plastids found in plants. Now their purpose we know it is primarily carrying out photosynthesis. Now in photosynthesis there are light reactions and dark reactions. So we will be talking when we come to photosynthesis some idea about light and dark reactions, but these chloroplasts they are responsible for capturing sunlight and helping the plant in manufacturing the food. When I say food, it is synthesis of carbohydrates. Then endoplasmic reticulum, we all know what function does it play in the cell. Endoplasmic reticulum, it is a connection between the nucleus and the cytoplasm, and it helps in what? It helps in manufacturing, processing, transportation of various proteins, fats, many molecules. Now there is smooth endoplasmic reticulum, rough endoplasmic reticulum. Rough endoplasmic reticulum is differentiated from smooth in terms of ribosomes, sites for protein synthesis. So it acts like a bridge between the nuclear, double-layered nuclear envelope and the cytoplasm. So it is a connection between the two. Now peroxisomes, what are peroxisomes? You will hear about peroxisomes many a times in plant metabolism. Peroxisomes can play a role where? Have you heard of something where peroxisomes play a major role? Which part of plant metabolism? So it takes part in photorespiration. Any idea what part of photorespiration? Peroxy. So we will take it up later. Photorespiration. Then there is, because of photorespiration, there is generation of lot of peroxy radicals. So plant has to have a mechanism of getting rid of, because then they are highly reactive species that can cause damage to cellular membranes and cellular components. So continuously the plant cell is working on to counter this damage. Then plasmodesmata. Plasmodesmata, plant cells, as I was mentioning, that they have a tendency to grow as aggregates. You will always find them in clumps. So one of the reasons is that plant cells are found to be attached to each other in many clumps generally. Why? Because of this phenomena. There are holes in the cell wall which you can clearly see through light microscope. These holes are called pits. They are the result of thinning of the cell wall during the plant cell division. When the cell wall, so the middle lamella is one of the part of the cell wall, all where the endoplasmic reticulum, because every component is divided into two. Now the endoplasmic reticulum, while dividing, does not get divided completely and gets compressed. Now these thinner regions then therefore become the continuity between the two daughter plant cells or the plant cells in connection. So now this continuity is not only just a simple gap, but it causes a live bridge between the two plant cells. So there is a crosstalk in terms of the endoplasmic reticulum and your, what else? Plasma membrane. So this is how the plant cells are found to be connected to each other. So this is called as plasmodesmata or plasma lemma. The continuity, the live bridge between the adjacent plant cells. Now plasma membrane. Plasma membrane is what? That you should be able to tell me. Plasma membrane is?
Generally we say it has a PLP structure.
What is that? PLP means what?
Protein lipid protein.
So, phospholipids and proteins. There can be some fatty acids. So, phospholipid molecules, and it is a bilayer phenomena. So, what is the use of plasma membrane? It acts like a semi-permeable membrane. When I say semi-permeable, which means that there is regulation of movement of molecules inside and outside the cell. This regulation can be active, which means, so where these transport proteins in the cellular membrane and the fluidity of the cellular membranes helps in the to and fro movement. Why is that needed? During metabolism, during plant growth or during survival, you may need to secrete out certain molecules, you may need to take up certain molecules. So there plasma membranes helps, and it also forms a barrier. Then ribosomes. Ribosomes, they are the site of protein synthesis. 60% is RNA and 40% is protein. Nucleus, we all know, it's a double-layered organelle. It is the brain center of the cell. It regulates all the different functions: cell division, cell metabolism, growth, then protein synthesis. All that is regulated by the, and what else? Nucleus is the site for genetic material storage. Then mitochondria, energy center of the cell. Now in plant cells, when light energy is not applicable, mitochondria is the organelle which caters to energy generation. In the presence of light, majority comes from chloroplast, your photosynthesis, ATP generation, and which is then utilized for, or reducing power even, so which is then utilized for synthesis of carbohydrates in dark reactions. Then vacuole. Vacuole, I said, is a distinctive feature in plant cells. Now, vacuoles you'll find, mentioned earlier also, that vacuoles in younger plant cells, you'll find they will be smaller in size and many numbers. But as the plant cell matures, grows in size, the matured plant cells generally have single vacuole all clubbed together, and they can occupy as far as 80% of the cell volume. So, turgor pressure, it causes the cell to maintain the cell turgor, or the vacuole when extends, it will prevent plasmolysis. The other added advantage would be, if there is good turgor pressure, then all the cell organelles will be extended and pushed towards the cell membrane. So, all the cell organelles, especially which organelles are the targets? Chloroplast, when sunlight is there. So that all the chloroplast is exposed on the surface and they can capture as much as possible the sunlight. Then she said storing molecules. Generally, what kind of molecules? It acts as degrading toxic molecules, degrading waste products, or it even acts as a site for storage of intermediate molecules for higher secondary metabolites, or even those toxic secondary metabolites which can be toxic to the plant cell itself. So that in case of a herbivory attack or a pathogenic attack, because the vacuoles are extended, big in size, they rupture and the secondary metabolite which is toxic is released. Otherwise, it is held. So now, anyways, the cell is now lysed. Why do you think it is happening? At least to prevent the neighboring cells or further damage. Then Golgi apparatus, this is also general in all eukaryotic cells. It is used for processing your shipping. So processing, when I say what kind of processing? For transport also, they might be needed to be conjugated with certain molecules. So that also happens in Golgi apparatus. And transport proteins or transport these conjugates will help it to cross the cytoplasm and be transported outside the cell membrane. Then, so now talking about the protoplast. What was protoplast? Protoplast was nothing but nucleus plus the cytoplasm. That is protoplast. Now distinctive features, what all distinctive features I told? One was the cell wall is composed of cellulose, hemicellulose, pectin and lignin. What is cellulose composed of? Cellulose is made up of, it is a polymer, polysaccharide made up of D-glucose. Simple chains. What kind of linkages? You must have read all this. I'm just revising it. Beta 1,6. Okay. Go back and read. Okay. So, beta linkages. One thing is correct. So, beta linkages, D-glucose units. That is cellulose. But if you see under some images of these cellulose, you will find that it is only can be observed under electron microscopy, and you will find it as fine thread-like structures, highly complex matrix, very multi-layered, and in each of those fibrils, I'll show you a picture, each of those fibrils themselves are composed of thousands of chains of D-glucose. So, that's the reason why cellulose is crystalline in nature and it is hard. Talking about hemicellulose, it is a heteropolymer. Now, heteropolysaccharide, heteropolymer in the sense, and it is amorphous in nature, weaker. Why? Because it is made up of many different kinds of sugar units. Xylose, then galactose. Find out there are others also. So, and it is amorphous in structure. That is hemicellulose. Now, pectin. That jam and jelly you people must be eating. fruit jam, fruit jelly. That jelly part and that gel-like structure is because I'm asking, so it is pectin. So this pectin gives that gel-like structure, and this pectin is again a heteropolymer which glues the plant cells and this plant cell wall different components together. Your middle lamella, which is above the primary cell wall, is very rich in pectins, and that is what helps. If it dissolves in water, if you bring water, it will gel. So that is what causes these plant cells to glue to each other. So then talking about plasmodesmata is another distinctive feature in plant cells. Cell-cell communication pathways. Chloroplasts are distinctive, which means plastids. Plastids are not only for photosynthesis or storage, but sometimes in secondary metabolism, you will find that they are site of synthesis of certain intermediates of biosynthetic pathway. If you'll see certain biosynthetic pathways like alpha tocopherol or vitamin E biosynthetic pathway, you'll find some part of the secondary metabolic pathway also is carried out in plastids. So not only storage, but also for site of synthesis. So cell wall, middle lamella, that's above the primary cell wall, which is rich in pectins. So those yellow threads, these are shown in the picture as pectins. And cellulose, if you see the primary cell wall is generally all the plant cells will have primary cell walls. They will have secondary cell walls are the ones which not all plant cells will have. Now this primary cell wall, what is the function of the primary cell wall? Is to provide protection and support to the plant cells. But it is still flexible in nature. Why flexible? Because these plant cells, the cells are still growing. Once they are young, they still need to grow. The cells are flexible to increase in size, to stretch, to become elongated. So the primary cell wall has the ability is more flexible in nature. So I have already spoken about the major carbohydrates in the primary cell wall. Cellulose under SEM looks like this. Complex fibrous, like if you have seen linen fibers or flax, your fabrics there. Now secondary cell wall is under the primary cell wall. It generally appears, its major function is to prevent the attack from herbivory and to give rigid support to the well-grown plant organ. So secondary walls appear which are richer in lignin. Now lignin you already must be knowing, it is very hard to degrade. Now precisely the reason is because hard to degrade, which means that its purpose in nature must have been to protect and support the full grown plant. So this secondary wall structure appears in cells which are already matured, and now higher specialized function is not the growth but to protect the cell. Then the secondary wall structure appears. So its major function is to protect the plant cell. Now cell wall openings, I said these are pits which are holes in the primary cell wall and through which the plasmolemma or the plasmodesmata passes through, which is composed of your plasma membrane and your endoplasmic reticulum. So these are live bridges. So this is how plasmodesmata is formed. It is formed when the portions of your endoplasmic reticulum get trapped across the middle lamella. You remember the middle lamella above the primary cell wall? As the cell is dividing. So what happens? The cell wall doesn't get made near these regions, or it is so thin that the endoplasmic reticulum, the plant cell wall, is then made in continuity with the adjacent cells, and those compressed regions of endoplasmic reticulum then become the continuity, or you call them as holes among the adjacent plant cells. Now vacuoles are very interesting organelles in plant cells. So we were talking about it helps in cell turgor pressure, maintain that turgor pressure. Then you said it helps in storage. It also helps in maintaining the cytoplasmic pH. So how does it help, do you think, in doing all these functions? Based off coming, selectively having some molecules stored in, then, so it has some degrading enzymes also stored in, which will be useful in degrading the waste product, degrading the toxic molecules or keeping the toxic molecules. How do you think all this is getting managed in vacuole, which is inside the cytoplasm?
Some enzymes.
Enzymes are doing what? What is helping the vacuole in carrying out all this function? Very nice. So it has a membrane called as tonoplast. So tonoplast is very rich in lot many number of diverse proteins. Now these proteins are transport proteins, then very different varied kind of transporters, can be simple protein through active transport, can be ion transporters, can be channel proteins. So there are many different kinds of proteins in this tonoplast, which is a single layer around the vacuole, which helps in the selective movement of the molecules from the cytoplasm to inner. So it contains water and all these salts inside. And the tonoplast is the one where through active transport, even and the electrochemical gradient is maintained. I said it helps in maintaining the pH of the cytoplasm. And you'll find that the vacuole pH is acidic in nature. Why do you think it is acidic in nature? And many of these enzymes, things which are present inside, they are active at acidic pH. Now plastids, there are very different kinds of plastids I was mentioning. Chromoplast are the kinds of plastid which can store your colored molecules. Then chloroplast for photosynthesis, leucoplast is for storage of many different kind of molecules, where they are further divided into amyloplast you must have heard, for starch. Then alloplast for oils, granules. Then proteinoplast for proteins. Now there are three different kinds of plant cells. One is parenchyma, then sclerenchyma and collenchyma. Now talking about parenchyma cells are the majority of cells present in the plant. They form the mass of the cells, the plant. Now parenchyma cells are those totipotent cells which we keep referring to. They form the mass of the plant and they are, for their entire life, they remain totipotent. Totipotent means they have the ability to organize themselves into a specialized function and again redivide and come back to totipotent stage and again redirect their metabolism to carry out another specialized function. So these are parenchyma cells. Generally, where will you find them? You will find them in all growing parts. In your photosynthetic cells, your spongy cells, then your mesophyll cells, which are there in your photosynthetic process in leaves. You will find these kind of cells. So the cell wall, they only have primary cell wall because as they are present in the young cells, which are growing, the plant cell wall, the size will keep increasing. There is change happening in the cell size, shape. So the cell wall needs to be flexible. So in all these parenchyma type of cells, you will find there is only primary cell wall, so that they are more flexible. You will find them square shaped generally, but still the shape can change a little. Collenchyma cells, here the cell wall thickens. So collenchyma cells are layered cells, which the role of these cells, they become a little more specialized. The role of these cells now is to support. So you will find them in regions where the growth is still happening, which means the root which is still elongating, growing, or the stem which is still elongating in the growing parts of the plant, so they are more elongated cells. So again, they are although lesser flexible than parenchyma, but still the cell wall is flexible. Now sclerenchyma cells, if you remember walnut, you must have seen walnut, then peach, such hard coat is there. And in your flax or in your linen, which are very difficult to tear apart. So these are sclerenchyma cells. Sclerenchyma cells appear when the cell has completely matured, there's no more growth happening. And at maturity, now the secondary wall develops, and the secondary wall, there's large amount of lignin in it. So because of thickness of the plant cell wall, and there is gradually, you'll find that the cytoplasm will be lost and they'll become vacuum, empty cells, empty chambers. So most of these cells, if you'll observe, they will be empty chambers with only cell wall. So generally their purpose is to provide structure and to provide protection against the external environment. So that is the sclerenchyma cells. So you will find them only in matured parts of the plant where there is no more growth required. So there are two types, fibers and sclerides. Fibers, I was mentioning, these are very long length, even up to 50 centimeters long cells. So like in your fibers, your fabrics, I said linen and flax. So then sclerides you will find they have much thicker cell wall, even more than fibers. And even small group of cells, you can see them together, or even single cells with large size is where they will be used to protect that peach pit and your what else? Walnut. Those are all your sclerides.